Welcome to my channel Adhyan with Flow. Let's start with prokaryotic cells. What is the prokaryotic cells? There are some examples like bacteria, they are prokaryotes. BGA, blue green algae, these are also prokaryotic cells. Mycoplasma, prokaryote. PPLO, this is also a prokaryote cell. What are the prokaryote means? Pro means primary level and karyote means they have less developed nucleus. Less developed nucleus. Yes guys, they does not have well defined organized nucleus. Okay, so if they don't have well nucleus, that means they does not have any kind of well defined genetic material. No guys, they have genetic material. Nucleus is not well developed, but doesn't mean that they don't have well developed genetic material. They have circular form of genetic material. Like us, we are multicellular. We have well defined cell, and inside the cell we have well defined nucleus. Inside the nucleus we have well defined genetic material. We call it as a DNA. Our DNA is double stranded, and their DNA is single stranded DNA, circular. Clear guys? Next, they are generally smaller and multiply more rapidly than eukaryotic cells. Means multicellular organisms are well defined, well organized, they are complex. But eukaryotic cells complex. But prokaryotic cells, they are simple in structure because they contain only one cell, guys. They are unicellular. They divide more faster than multicellular because they are simple, but they are complex, organized. So they take lots of time. This is the cellular form. It does not require tissue level, organ level, body level system. They don't have this kind of system. So they can replicate, they can divide multiple times. I know they divide, but there is drawback. Their lifespan is less. Lifespan means their life duration. But these are complex. We know that we are complex. We have at a particular age, when we young and adult, when we reproduce, we produce 1 to 2 progeny. But these progeny span is high. They may vary greatly in shape and size. Yes guys, they are single cell structure. Okay, they have single cells, so they can be arranged in another multiple form. It may be rod like structure, example is bacillus. It may be spherical, example is coccus. It may be coma shape, example is vibrio. It may be spiral, example is spiralum. Look at here, I think. Oh no, there is no diagram guys. There is no, look at here, PPLO diagram. Clear guys? Compare this cell and compare this cell guys. What you get? Their size is less than our organelle. Okay, now the organization of prokaryotic cell is fundamentally similar, even through prokaryotic exhibit a wide variety of shape and function. They want to say that they may be vary in their shape and size and structure, but their function is similar. Prokaryotic have a cell wall surrounding the cell membrane, except in mycoplasma, and this thing is important. They said that prokaryotic cells have cell wall, but if we talk about animals, then it does not have cell wall. But if we talk about plants, they have cell wall. Next, the fluid matrix filling. Okay, this is the cell, their body, and they are rich in their cytoplasm. Our cell also contains cytoplasm guys, not difference. There is no well defined nucleus. I already told you. The genetic material is basically naked, not enveloped by nuclear. Guys, nucleus is not well developed, so how can you expect that nuclear membrane will form? Guys, this is the cell, and this is not a specific in structure. They does not have perfect nucleus guys, so how can you expect that nucleus and membrane will present? How you expect? Genetic material is also not developed, but they have circular DNA. Look at now guys, I already clear all the terms. Single stranded DNA, circular in form. Look at here. These small DNA are called as a plasmid. These are circular DNA. We call it as a plasmid of prokaryotic cell. The plasmid DNA confers certain unique phenotype character. These are important guys. When we study biotechnology, they are huge importance. They also work as a vector. Okay, one such character is resistant of, yes guys, I told you now, when we study biotech, there are huge important of plasmid. I know this is not your in syllabus, but if your further classes, when you pass 11th and you come in 12th class, you can face biotech. Okay, this is important. Plasmid are very important in biotechnology. Okay, nuclear membrane is found in eukaryotic. Okay, we will discuss in eukaryotic this thing. Cell envelope and its modification. Okay, we know that prokaryotic cell have cell envelope, means they have plasma membrane and they also have cell wall. This thing is clear. Now what they want to say, that their outermost covering, the cell envelope consists of tightly bound 3-layered structure. We call it as a glycocalyx. They want to say their cell, okay, single cellular structure, but they made up with 3 layers. The first layer is glycocalyx. I'm writing here glycocalyx. Okay, this layer is cell wall, and this layer is EM. Look at here, this and this. Alright, and according to their staining process, some bacteria are gram positive, they show positive response for staining, and some are gram negative, they does not stain properly. Okay, now next, glycocalyx different in composition and thickness among the different bacteria. This outer layer may be vary according to bacteria. This layer, glycocalyx layer, vary in different bacteria to bacteria. Okay, it could be slime layer. Look at here, na, glycocalyx different in composition and thickness among different bacteria. So they may be vary. Glycocalyx may be vary according to bacteria to bacteria. It may be slime layer. What is slime layer? Look at here. It could be loose sheet called slime layer in some bacteria, while the other it may be thick and tough, and it may be thick and tough. We call it as a capsule. This is the loose, and this is the thick and tight, rigid. Okay, the cell wall determine the shape. Of course, cell wall, they give strength, they give support, they make it stronger. Okay, the plasma membrane is selectively permeable. According to our plasma membrane, this is also same. The plasma membrane with made up with lipid bilayer. They made up with lipid bilayer. Plasma membrane with outer layer and inner layer. Lipid bilayer. Now they are selective permeable. What they do? This is the outer plasma membrane. This is the inner side. If some particle goes inside the cell, so this selectively permeable towards this molecule. If this plasma membrane will allow, then they can easily come. If they don't allow, then they can't come. Some special membrane structure is mesosome. There is some invagination of plasma membrane. We call it as a mesosome. What is the special function of mesosome? Okay, look at it. Which is formed by extension of PM into the cell. The extension are the form of vesicles. It may be vesicles, tubules, and lamellae. Okay, they may be vesicle shape, they may be tubule shape, and they may be lamella. They help in cell wall formation. What are the use of mesosome? They are helpful for cell wall formation. DNA replication and distribution of daughter cell. They also help in respiration, secretion process, and increase surface area. Mesosome are also helpful in respiration and some exchanges of molecule, some enzymatic exchange of molecules. So mesosome, I'm writing here. Mesosome help in respiration in bacteria. Okay, DNA replication in bacteria. Remember my thing. Some secretion process, if they want to secrete something, they increase surface area. They're responsible for enzymatic activity. Clear? In some prokaryotes like cyanobacteria, there are outer membrane extension into the cytoplasm called chromatophore, which contain pigments. If outer membrane inside, we call it as a chromatophore. Clear, guys? What they contain? They contain their pigments. They synthesize their food, so they can store their pigment chlorophyll. Okay, na? We say na pigments, they may be chlorophyll, guys, because they synthesize their own food. Bacterial cell may be motile or non-motile. This cell is motile or non-motile. According to what? If they have flagella, they can motile. If they have cilia, they can motile. What is the structure of flagella? Structure of flagella are divided into 3 parts: filament part, hook part, and basal body. And this is the filament part, and this is the hook part. This is the basal body, there is a filament, and this is the hook part. They also have pili and fimbriae. Okay, now here is the cell organelle. Ribosome. What is the function of ribosome? One of the most important function is protein synthesis. Remember this function. In multicellular, they are ATS. ATS means Sydberg unit. And unicellular, you can say prokaryotes and eukaryotes. ATS, 70S. This is clear? S is the Sydberg coefficient. They break into two subunits, large subunit and small subunit. They also break into large subunit and small subunit. 70s further divided into 50s and 30s. 80s again divided into 60s and 40s. Remember this ratio. This is important. Remember, always remember this ratio. They further divide, guys. They further divide into small subunit. Means there are some large unit and small unit. Large unit and small unit. This is 60s, this is 40s, this is 50s, and this is the 30s. Clear, guys? They also break down into 5.8s, 5s, 80s, 16s, but this I'm not going deeply inside. Okay, there are some including body in our cell. Including body. Reserve material in prokaryotic cells are stored in the cytoplasm in the form of inclusion body. If bacteria want to store some food, if they want to store some food material, some pigment, anything else, they make small body in cytoplasm. And they store their pigments, they store their food, they store their oil, they store their water, they store their pigments. Okay, or they are free in cytoplasm. They are not specifically binded, covering with a high profile. Come on, guys. Now, next is eukaryotic cells. We complete our prokaryotic cells. Now it's time to study about eukaryotic cells. Eukaryotic include protists, plants, and animals, fungi. And what kind of example is including in prokaryotics? Wait. Where prokaryotic? Where? Yes. Bacteria, BGA, mycoplasm, PPLO. And eukaryotic example is protists, plants, animals, fungi. Yes, specifically kingdom, guys. Protista kingdom, plant kingdom, animal kingdom, fungi kingdom. Eukaryotic cells, there is extensive compartment, means overall they want to say that they have specific cell. Every cell contain according to their cell organelles, and these cell organelles are well defined, covered with fully established membrane. They have perfect structure, well organized cell membrane, cell wall. Okay, they have they have contain all kind of cell organelles, mitochondria, chloroplast, ribosome, everything, microtubules, everything they contain. Okay, all eukaryotic cells are not identical. Plant and animal cells are different. Of course, they are different. This is the plant cell and this is the animal cell. Wait, this is not a beautiful. This is the animal cells and this is the, why I'm drawing this rectangular? Why? Because they covered with cell wall and this is only covered with TM. That's it. And they also contain nucleus, they contain mitochondria, they contain chloroplast, they don't contain chloroplast, they contain lysosome, they contain lysosome, they contain peroxisome, they contain peroxisome, they contain ER, they contain ER. They contain microtubules, they contain microtubules. They don't contain centriole, they contain centriole. They contain large size vacuole, they contain small size vacuole. They contain 70s ribosome, they contain 80s ribosome. Guys, oof. Okay, now clear? Look at here, guys. Look at why I suffer. This is the well diagram of the plant cell, and this is the animal cell. Look at here, guys. I'm describe only one thing. These are rough ER. Why we say rough ER? This is the endoplasmic reticulum, and they contain small ribosome. They are helpful in what? Protein synthesis. There is nucleus. Inside the nucleus, they have nucleolus. This is the Golgi apparatus. This is the nuclear envelope. Look at here, this outer covering. This is the plasma membrane. This is the large size of vacuole, store food, functioning for storage. This is the cell wall. It may be made up with cellulose, hemicellulose, pectin. We discuss later in your NCERT. They elaborate it. Don't worry. This is mitochondria. What they do? Produce ATP. These are ribosome. Plant cell also contain ATS kind of ribosome. Here's difference between plant cell and animal cell. Guys, this is also contain ATS. This is also contain ATS. Okay, but their mitochondria contain 70S. Their chloroplast, where is chloroplast? They also contain 70S because these two organelles come from outside them. This is not a part of our cell. They are come outside. Chloroplast come from the outside and mitochondria also come out from the outside. They contain microtubules, they contain plasmodesmata. Only plant cell contain plasmodesmata, not animal cell. And last is smooth endoplasmic reticulum. Guys, this is smooth. They does not contain any kind of ribosome. So they are not useful in protein synthesis, but they are useful in storage. They store. And lysosome, they contain many kind of enzymes. Peroxisome, they also contain enzymes. This is the what? This is the animal cell, guys. This is the microphylla. Increase the absorption area, increase the surface area. This is the plasma membrane. They does not have cell wall. They have centrioles, but the plant cell does not have centrioles. They have peroxisome. They contain enzymes. Lysosome, they also contain enzymes. These are necessary, guys. We can also call it as a suicidal bag. Okay, ribosome, they are useful for protein synthesis. If ribosome present in endoplasmic reticulum, we call it as a rough. Okay, they useful in protein synthesis. If ribosome not present in ER, they useful for storage and cell signaling and protein sorting. Clear, guys? Mitochondria useful for ATP formation and they contain 17S. You don't like that 70S? This is present in prokaryotic cells. So some scientists say these are bacteria, and throughout the evolution they insert in our cell and they give ATP to us. Nucleus, well defined nucleus, guys, well defined. Here is nuclear envelope. Inside the nucleus they have nucleolus. What is synthesized? They synthesize RNA. Now, guys, this is the structure of animal cell, and here this is the structure of plant cell, and I already described what is the eukaryotic cell and prokaryotic cells, guys. In our next video, we will discuss one by one, starting with cell membrane. Okay, beyond your NCERT. Okay, so let's meet in next video. Then, thank you so much, and subscribe my channel Adhyan with Flow.